Want to say hi to everybody here who's joined. We're going to give it a few more minutes before we start to allow more people to join the webinar. AG, why don't you go ahead and start with the introduction?
Yeah, so just 1 minute. Can you see my screen?
Yes.
Yeah.
So,
Yes.
hi everyone. Thank you everyone to join us today. This is our June webinar promoted by the Pharmacology Education Initiative, which is a new collaborative program launched by the Pharmacological Research Family of Journals, together with the Elsevier and the International Union of Basic Clinical Pharmacology. My name is Deidiane Elisa Ribeiro. I'm the coordinator of the Pharmacology Education Initiative, and I would like to briefly present this project to all of you. So the main objective of our program is to provide and disseminate the free access to high-quality content on pharmacology for students and researchers worldwide. To achieve this goal, we are promoting a series of webinars on methodology in basic and clinical pharmacological research over this year. And then we started it some months ago, and today we have the pleasure to have Dr. Cheng. But please be tuned to our next webinars. And of course, that nothing of this would be possible without the amazing team working on the Pharmacology Education Initiative. Along with us, we have the full support of the editorial board of the Pharmacological Research Family of Journals, particularly Emilio and Anne-Marie, who have guided us through every step in this journey. We also count with invaluable collaborators, as I said before, which are Elsevier and IUPHAR. So please, for more information and updates, join us on LinkedIn. And after this webinar, you can download the personalized certificate of attendance on the website, putting this code on the screen. And without further ado, I would like to invite Anne-Marie to speak a little bit more about the program and our initiative.
Thank you, everyone. Thank you, Deji. I'm Anne-Marie Pordone. I am the publisher, one of the publishers for Elsevier for the pharmacology titles, and I'm really happy that you were able to join us here today. As Deji said, today we have Dr. Cheng, who's going to give a lecture for us. And then also I'd like to introduce Huarong Xu, who is going to moderate the lecture today. So Huarong, if you can proceed with the introduction.
Okay, Thank you. Hello, everyone. So this is Huarong from Shenyang Pharmaceutical University in China, and it is really a great honor to introduce a very famous young scientist in the field. We call it pharmaceutical analysis in China, and who is a professor and Dr. Cheng Yan. And Dr. Cheng was graduated in pharmaceutical science from Kyoto University in Japan. And she is now an associate professor of pharmaceutical analysis department in West China School of Pharmacy in Sichuan University. And she is also a visiting associate professor at molecular imaging program at the School of Medicine in Stanford University. And Dr. Cheng is she specializes in molecular imaging in clinical precision diagnosis and drug development, which includes multimodal imaging modalities to monitor molecular events in living subjects, thorough diagnostic reagents, and targeted imaging probes, and imaging-based pharmacokinetic approaches. And she has participated in the development of several diagnostic radio tracers into clinical translation and has granted over 20 patents. And the speech Dr. Cheng will give us today is molecular imaging of neurodegeneration and its application in defining drug efficacy through biomarkers. So let's welcome Dr. Cheng's speech.
Thank you and welcome all of you online. I'm very happy to be with you today, and I especially want to thank our webinar organizers for their hard work in getting us together so we can help each other think. So can you see my slides?
Yeah, looks good.
Okay. The title of my talk is Molecular Imaging of Neurodegeneration and its Applications in Defining Drug Efficacy through Biomarkers. So I'm just going to get into the slides. Briefly, the outlines for my talk, which will take about 45 minutes, will cover introduction to molecular imaging, a quick overview of neurodegenerative disease or NDDs and neurodegeneration. Then we'll talk about the identification of neurodegeneration biomarkers using molecular imaging approaches and their applications in defining drug efficacy through biomarkers, which data will be primarily based on Alzheimer's dementia. And to finish up, hopefully with a little bit of a discussion on challenges and opportunities about what we can do together as a field moving forward. So I'm going to start with introduction to molecular imaging. Many important biological processes occur deep inside the body. So to study biological process in vivo requires new technologies to understand functions at single cell level. Besides the classical approaches, the term molecular imaging is defined as the in vivo characterization and measurement of biological process at the cellular and molecular level through the use of imaging device. So there are various imaging modalities, including the most common medical imaging modalities like the X-ray imaging, the computed tomography or CAT scan, the MRI, the magnetic resonance imaging, the PET, the positron emission tomography, or the SPECT, the single photon imaging computed tomography, and also with the preclinical imaging modalities such as the micro-PET, micro-MRI, and also small animal optical imaging systems. So molecular imaging probes the molecular abnormalities that are the basis of the disease rather than to image the end effects of these molecular alterations. So unlike the conventional anatomical and the functional imaging that focus on anatomical structures, molecular imaging allows observation and tracking of specific molecules, proteins, or cellular process in real time within the body. This ability of molecular imaging promises improvements in specificity and quantitation and offers unprecedented opportunities for early disease detection, personalized treatment planning, and monitoring of therapeutic response and/or treatment follow-up. The main advantage of molecular imaging is its ability to characterize pathologies of disease tissues in vivo without invasive biopsies or surgical procedures, which makes its primary importance in applications for tumor and brain disease such as neurodegenerative disease. So the neurodegenerative disease or NDDs are a heterogroups of neurological disorders adversely affecting the lives of millions of people worldwide and entails the progressive loss of neurons in the central nervous system or peripheral nervous system. The most common NDDs here on the right side include the top two, Alzheimer's disease and Parkinson's disease, almost over 19% of all NDDs, and others like Huntington's disease. So NDDs are progressive disease featured with a complex multifactorial process that causes neuron death in the brain and in the spinal cord, and the process is called neurodegeneration. So the neuronal subtypes are differentially affected in NDDs, like for Alzheimer's disease here and here, they have the cortical and the hippocampal neurons affected. And for Parkinson's disease and Huntington's disease, they have striatal neurons affected. So in this slide, neurodegeneration is accompanied by progressive process happens mostly in the brain, such as abnormal protein aggregation, which we will discuss later for the most, and oxidative stress, neuroinflammation, external transport deficits, and other pathophysiological process. So for the primary one, the pathological protein aggregation, for various NDDs, abnormal protein misfolding, aggregation, and deposition in the brain have been considered as the pathological hallmarks of these NDDs. So NDDs have different kinds of protein clumps in the brain and spinal cord. Misfolded protein that build up and clump together are thought to be the primary reason for these disorders. The proteins here, they can clump together to make, like here, amyloid beta plaques. They can be extracellular, and like the tau tangles, they can be intracellular inclusions. And also here, at like the Parkinson's disease, for the alpha-synuclein plane, and the Huntington's disease, they have aggregated Huntington to form the beta sheet inclusion bodies. What we know for sure is the hallmarks of Alzheimer's dementia, as described by Dr. Alzheimer over 100 years ago, that it has these extracellular aggregations of A beta plaques and intracellular tau tangles. We know there's a lot of genetic that also points towards amyloid. So for amyloid and tau PET imaging has demonstrated the clinical utility, provide tools to evaluate the disease stage, improves the diagnostic accuracy, and monitors the efficacy of interventions. Especially, amyloid PET is expected to have greater utility after the approval of anti-A beta immunotherapies, such as lecanemab and aducanumab by FDA, which we will discuss in later slides. So future directions in molecular imaging of neurodegeneration are best described and illustrated by the trends in imaging Alzheimer's dementia, for which the most advanced imaging tracers specific to the neurodegenerative process have been developed. As the genetic and mechanistic kinship among these disorders is becoming progressively clearer, much of the imaging that is applied to Alzheimer's dementia can similarly be applied to the study of other NDDs. So next, we will talk primarily based on the data from Alzheimer's disease. So in slide 11, this slide represents what we believe the essential model of Alzheimer's disease. The first 100 years we've been looking at the disease, we've been focusing on everything all the way at the very right side of the screen, where people are already firmly in dementia land. And what we realized, particularly looking at families with these altered dominant genetics and doing cognitive testing on them every year, is that up to 25 years before they have any memory impairment involvement. We see here on the left, we see this red line start to come up, and this is abnormal A-beta amyloid. And we can detect this. We can detect this with amyloid PET scans. So 25 years before they have symptoms, there's something molecularly wrong. This then continues to build, and in some way, that's where the hand waving. We're not really sure how that happens. This tends to occur somewhere between 10 to 15 years that we can detect the earliest before people start to have memory problems. Soon after that, we can see the green line start to rise, which is where we start to see actual structural change. And this is like high magnet susceptibility on MRI. And we can see in both brain volume change or on FDG PET. And then you start to get some memory problems and you start to reach into the MCI range, which means mild cognitive impairment. And then late that progresses, then you start to lose functional status, and that's when we start calling people dementia. So really the last 10 to 20 years, instead of focusing all the way on the right, we've been focusing a little bit closer to the line between MCI and dementia, in hope that if we get people earlier, we might be able to start the intervention. So now there's some debate about the amyloid hypothesis. In particular, there's a lot of data showing that clinical pathological correlation between these proteins forming in the brain and memory loss is not linear. So it brings the question, is that the amyloid hypothesis is the only thing? Or are there more things? And the answer for now is very complicated. So these days, there's a lot of talk about the inflammatory hypothesis. It points towards inflammation potentially having a role. There's been a lot of other studies showing that. And particularly these days, with all the patients getting COVID, and suddenly getting memory loss, and then seemingly suddenly getting diagnosed with Alzheimer's dementia, as well as other virus sort of pointing towards increased risk of Alzheimer's. So this slide shows the schematic presentation of expected paradigm shift in how Alzheimer's dementia will be defined, diagnosed, and treated in future, together with the role of molecular imaging, especially PET scan. Before, definitive diagnosis of Alzheimer's has relied on autopsy, which is confirmed by the presence of protein deposits in post-mortem brain tissues. Of course, it's too late. But for the potential patient, AD patient, the evaluation of person with suspected memory impairment includes a comprehensive set of symptomatic assessments, including a detailed medical history, physical and mental status examinations, and neuroimaging studies. For neuroimaging, such as MRI is being used for a long time in the clinic in patients with slowly progressive cognitive impairment leading to dementia. So the MRI simply inspects images without the need to quantify them. However, the brains of Alzheimer's cause subtle changes on MRI, but remarkable changes on PET scan, like using the metabolism, amyloid beta, or tau PET tracers. So the landscape of imaging Alzheimer's dementia is changing very rapidly. In 2012, fluorobetaper was first approved by the FDA for routine clinical PET imaging of brain A beta deposition, and this tracer was then approved by the European Medicine Authority, or EMA. So slowly, these tools are becoming part of the clinical armamentarium to define the presence of early Alzheimer's change in patients with MCI, or to differentiate AD from other dementia. So next, we'll talk about the application of how PET scan works and how to develop PET tracers for Alzheimer's disease. So positron emission tomography, or PET, is a quantitative molecular imaging technique that allows in vivo analysis of neurotransmitter systems in the brain by detecting gamma ray photons resulting from annihilation of positron-emitting radionuclides. So the PET signals come from positron-emitting radionuclides. Of course, the body naturally doesn't have any radionuclides. So to get specific signals to detect the biomarkers in the body, we have to answer the key questions below, including which reliable biomarker to select in the body, and how to develop a specific molecular probe for the biomarker. And as the biomarkers are mostly present in the brain for NDDs, so the development probes has to efficiently cross the blood-brain barrier, or BBB, and enter the brain. This slide shows schematic presentation of general procedures of PET imaging. The positron-emitting radionuclides are obtained from cyclotron. For radionuclides with short half-life, such as carbon-11 with 20-minute half-life, they are mostly obtained from on-site cyclotrons. So next, chemistry and radiochemistry are applied to prepare for the radio tracers between precursors and radionuclides. Then the formulated radiopharmaceuticals are administered into the body for PET scan and data acquisition. As PET imaging enables the quantification of the physiological parameters of interest in single or multiple organs or parts of the body right after the injection of radio tracers into patients or animals. So PET scan not only obtains non-invasive and dynamic image data, but also enables quantitative analysis. To show all the viewers, so here's a video of three-dimensional PET imaging we did of the whole body of a normal mouse on from a micro-PET CAT scanner after intravenous injection of fluorine 18 labeled radio tracer. So generally, we can see the structural information was obtained from the quick CAT scan on the imaging device first, and later the PET scan was performed So here we can see the whole body, and there are signals, passive radio signals in the brain, and also here in the liver, and some in the kidney, not very obvious. And as we can see here, we have all the structural information and with the molecular information. The images were obtained by image reconstruction. So we cannot get the PET images immediately after the imaging. So this slide shows the development of radioligands for PET imaging of A-beta deposits studied over the last decade. So we published this review paper in 2023. So we summarized all the radionuclide, radio tracers for A-beta deposits. For efficient penetration of the blood-brain barrier, the ability to enter the brain and specifically bind to A-beta deposits are essential requirements of small molecular radioligands. So we can see here as the time, like for 2 decades before, a variety of compact radioligands have been developed as potential PET tracers on the basis of the structure of mostly applied dye for staining protein aggregates that's called thioflavin T and Congo red. So based on that scaffold, here the neutral benzodiazole analog called Pittsburgh compound B was the first selective probe for A-beta deposition and have been most extensively studied for in vivo PET imaging of A-beta plaques in living individuals before clinical symptoms. So here's a typical amyloid PET scan with tracer, the carbon-11 labeled Pittsburgh B in cognitive normal, MCI, and Alzheimer's dementia. So compared from the images of the cognitive normal participants, the carbon-11 Pittsburgh B PET images on MCI and AD patients displayed moderate to severe binding throughout cortex, respectively. However, after the PIB, due to the short half-life of PIB of 20 minutes, that's very hard for the radiochemist to do the labeling of PIB. So more tracers with another radionuclide called fluorine-18 with more suitable half-life of 110 minutes, almost 2 hours, have been developed. So here I'll take this compound, the FPYBF2, as an example to take a glance at how to develop a PET tracer for A-beta deposits in the brain. So FPYBF2 is fluorine-18 labeled radio tracer we've previously developed for A-beta deposits. From the structure, we can see it is a very compact molecule with molecular weight less than 500 Dalton, which will facilitate efficient brain uptake by passive diffusion. The tracer preparation mainly consists of 2 parts. For this tracer, it is obtained from organic chemistry and was labeled using radiochemistry. So first, the precursor for fluorine-18 labeling was first prepared by organic synthesis. So it's several steps. So then the radio tracer was prepared by radio tracing of precursor with fluorine-18 under heated conditions. So next, the radio tracer was evaluated by in vitro autoradiography using transgenic mouse brain and human brain tissues with AD, Alzheimer's dementia, and the normal control. So in both types of brain tissues confirm specific and selective labeling. and binding of A beta deposits. And with later biodistribution data, which was not shown in this slide, displayed efficient brain uptake and washout from the brain with time in mouse models, shows the potential of this tracer for clinical translation. So next, in slide 22, it shows a clinical study of our developed tracer in cognitively healthy volunteers and Alzheimer's disease patients. So from the PET scan images of healthy volunteers showed that the tracer uptake was mainly observed in cerebral white matter, while AD patients showed tracer uptake in cerebral gray matters as well as cerebral white matters. So later, the time activity curve, or TAC, shows the signal dynamics in different brain regions, like including the cerebral, the cerebella, the thalamus, and the pons. So SUV here on the left stands for the standard uptake values, which is the most widely used measure for quantifying amyloid burden. It is a simplified method based on computing the ratio of tracer uptake between a target region and a reference region in PET acquisition, when the radio tracer is expected to have reached the equilibrium. In the evaluation of a 17-minute dynamic PET-CAT scan, the TAC, the time activity curve for the cortical accumulation, was calculated in 13 volunteers at the same instant, and the SUVs of cortex showed the equilibrium phase with a similar level of that of cerebellar cortex at 15 to 17 minutes after injection of the tracer. So they came from this curve, they're almost the same. So the TAC, on the contrary, the TAC, the time activity curve for the cortical accumulation of AD patients showed much higher here for the cerebral cortex accumulation, retention, and reached the higher retention level than that of the control, the reference cerebellar cortex. So based on this, in clinical trial settings, quantification may be used to identify the optimal window for therapeutic intervention. So over the past decade, many studies have demonstrated the level of analytical and clinical validity of amyloid PET in routine clinical use. So global multi-center studies aiming to unravel the influence, prognostic value, and role of amyloid deposition in AD timeline have been ongoing for some time. And the use of different amyloid PET tracers grew in both clinical and research settings. There was a need for inter-tracer standardization of the SUV metric in multi-center collaborations. To this end, here the centiloid or CL scale was developed, which is an unbounded zero, means gray matter signal of young healthy controls, to 100 typical AD patient signal scale that conveys a single patient's amyloid burden based on 2 anchor points using the PIB, the Pittsburgh B SUVs. Universal cutoffs, or the threshold values to denote amyloid status, can be applied alongside visual rays and in longitudinal multi-center studies to facilitate intercenter and intertracer comparisons. So here in this slide, various centiloid thresholds established in the literature and currently in use for clinical trial inclusion are summarized. So this can be further be illustrated by a HEAD 3-2-14-5 study, which is a phase 3 clinical trial started in 2020 for anti-A-beta antibody, BAN 2401. So this study requires participants to have specific levels of amyloid pathology, either intermediate with 20 to 14 centiloid or elevated over 14 centiloid as elevated A-beta. The centiloid scale has been used in clinical trial settings to track therapy response measures, determine strategies for reducing AD prevention trial sample size, and improve patient selection for trials, and could assist in treatment endpoint decisions. So next, we'll see how molecular imaging are being applied for disease detection, personalized treatment planning, and treatment follow-up. So we really believe in amyloid hypothesis. So anti-A-beta immunotherapies have been developed for a long time. The first generation of immunotherapies for A-beta deposits were these antibodies, and unfortunately, I'm really bad at pronouncing, and so I'm not even trying it. But largely, the first one targeted all forms of amyloid, and this was not effective, either a biomarker level or a clinical. So the middle one targeted more soluble monomeric forms of amyloid again. This one was not effective. And then the bottom one also targeted A-beta, but none of them were effective. So in this slide, let me get to the second generation of the therapy, and this is where things started to shake a little loose and get a little confusing. So the second one, this one showed that it was able to clear amyloid in patients and patient brains based on amyloid PET scans. Clinical impact was pretty much negligible, and it actually didn't do a good job as clearing amyloid as some of the other antibodies that on the list. So the third one, the aducanumab, it does a really good job of clearing amyloid from patient brains based on the amyloid PET scan. Clinically, it has not shown to have a definitive impact. There's sort of trend, but it doesn't quite reach statistical significance. This has accelerated FDA approval. So next, we get to the lecanemab, which targets the soluble aggregates with oligomers and protofibers. So for the initial phase 1 study was actually published in 2016 and was a pretty small study. It targeted patients that either mild or moderate Alzheimer's disease, dementia, and out of 16 patients who were given this medicine, 3 of them with 3 weeks developed amyloid-related imaging abnormalities. And interesting enough, we actually saw that in 2 of 20 people on the placebo arm. So the phase 2 study was actually published in 2021 and listed almost 900 patients that had either a mild cognitive impairment or mild Alzheimer's dementia. It used 5 different doses across 18 months. Their primary endpoint was to see if there was a reduction in the clinical dementia rating scale by at least 25% at 12 months. They did not meet that year, but they did meet some other secondary endpoints, and those included reduction in amyloid PET at 18 months. So for the phase 3, multi-center, multinational, double-blind, randomized control study was performed in almost 215 global sites in multiple countries, including United States, Australia, Canada, China, Singapore, Japan, Spain, Sweden, France, Germany, Italy, and United Kingdoms. So the stratified with longitudinal amyloid and tau PET scans and CSF analysis. Now, what excites us more is that in the secondary studies, where in the figure on the right shows that the monoclonal antibody that targets amyloid reduce amyloid. So we could have guessed that was going to be the case. So next, beside the above imaging modalities ready for clinical use, molecular imaging can also be applied for preclinical use, such as micro-PET, microspect, micro-MRI, or small animal optical imaging and photoacoustic imaging systems. We don't have enough time today for a comprehensive introduction of all modalities, so I'll just take a very quick view of optical imaging for detection of Alzheimer's biomarkers in the brain. So similar to other molecular imaging modalities, it is critical to obtain specific signals or biomarkers. So here is especially for fluorescent imaging. So a molecular probe is developed and administered into the animal before putting it into the imaging device for imaging. Here from a study from from Harvard, this slide shows a fluorescence probe targeting A-beta deposits in the brain. So non-invasive imaging of the transgenic mice containing APPPS1, so gradually have the A-beta deposits in the brain, and normal controls shows that distinct signal difference from the images. And later, from the image analysis, the semi-quantitative analysis further confirmed that the longer retention in the APPPS1 transgenic mice brains than in control compared to the retention in the control mice. And then after that, the probe was then applied to non-invasively monitor the therapeutic response. So they administered the therapeutic drugs into the mice and then injected through the tail wing with the fluorescence probe and can see the change in the brain in real time and non-invasive way within the same mice. So for the last of my talk, I would like to finish up with some discussion on challenges and opportunities of molecular imaging for NDDs. So first, challenges. First, more tracers are in great need for protein aggregation pathology imaging, such as the alpha-synuclein in Parkinson's brains. So we still don't have enough tracers for the NDDs. There's a lot of the large part of the different kinds of NDDs, and we still lack the effective and selective the probes. So in our group, we are now focusing on developing alpha-synuclein probes for the Parkinson's brains detection. So in specific NDDs, more than one protein opacity is often present, and conversely, specific protein pathologies are often co-found in different NDDs, such as tau, which is present not only in AD, but also in certain forms of frontotemporal dementia and some other kind of movement disorders. So second, the role of imaging neurotransmitter system will be extended. So like the first one, in NDDs, the degenerative process may affect all kinds of brain tissue components. So all the many neurotransmitter systems can be altered in this disease. What we have introduced the PET scan and the SPECT scan, the nuclear medicine are the only imaging techniques capable of in vivo tracking of the state of these neurotransmitter systems and of respective drug effects. So these imaging techniques play a very important, significant role both in clinical routine and for research. So third, new technologies for molecular imaging of NDDs will be applied in the future. Although not much mentioned in my talk, the application of new image acquisition and processing technology is anticipated to improve and broaden the application of molecular imaging in NDDs as well, such as hybrid PET MRI technology allows the simultaneous evaluation of both molecular and structural or functional changes in the living individuals. And also, advanced image acquisition and processing protocols can also benefit the molecular brain imaging and with the artificial intelligence, like for the image reading. So last on my talk, the opportunities, molecular imaging will become the standard for diagnosis of NDDs. It will also become the decisive imaging test for experimental and clinical drug application. Longitudinal molecular imaging is an ideal tool to assess regional treatment effects directly within the brain. Future scenarios are imaginable in which serial imaging allows individual tailoring of treatment intensity and duration. Another perfect example of personalized medicine. Furthermore, molecular imaging will redefine its general role with regard to fluid biomarkers to diagnose neurodegeneration. The answer potentially depends on what kind of PET readout is obtained for and for which purpose the diagnostic test is performed. Besides, PET imaging will also serve as the gold standard in vivo test against which novel fluid biomarkers are widely dated. So, and I think with the above, I'm going to wrap things up and see if anybody have any comments or questions here. And thank you all for your patience. Thank you.
Anne-Marie, I think you are muted.
There. Sorry. Thank you. Thank you so much. Huarong was supposed to moderate the chat, but he's having some technical issues. So I wonder if somebody else could ask the questions that have come in the Q&A. For everyone attending, please don't raise your hand. You can't use your camera or your microphone. You need to put the questions into the Q&A.
I'm open the Q&A and I can make the questions. Meanwhile, maybe I could also make a question.
Yes.
Dr. Cheng. So thanks so much for explaining presentation. It was very, very spark.
Deji, your connection is very bad. We can't hear you.
Oh, God.
So if any of the participants want to put questions into the Q&A, we'd be really glad to take your questions there. Samia, you have a question?
Yes,
Hi.
so while we wait for the participants to be less shy, first of all, thank you very much. Was really, really interesting and very clear presentation with very important insights. I think for everyone working with neuroscience, like beyond neurodegenerative disorder. So it was really interesting. I was thinking in a situation, for example, where you want to combine the evaluation of different biomarkers. I know that it's common, for example, that you can do, let's say, PET imaging, later on you do autoangiography to do the assessments. In your experience, what would be the best approach to have an overview, for example, of disease development in this case when you want to assess more than one biomarker in the brain?
Thank you for the question. So this is also a key question for the detection of NDDs brains because they have more than one protein, maybe they coexistent in the same brain. So now the selectivity of a probe is very important. And in addition, we have the multimodal or the bifunctional probe or multifunctional probe that they can target the different biomarkers at the same time. So that's our goal. Also, because Alzheimer's is the they have developed the very large amount of tracers. So now in clinic practice, they also use different tracers in the same patient, like for A beta and tau tracer. So because fluorine for carbon 11, it's a 20-minute half-life. So they use the PIB first for the patient to see the A beta, and then they will use a tau tracer because the FDA have approved A beta and tau tracers now. So they can do the PET scans during one day. So the patient can go to the clinic or the hospital, the facility to get the first, for example, from the morning, they can get the PET scan, inject the Pittsburgh B to get the A beta PET scan, and then later in the afternoon, after the second one, to inject into the body and to get the tau PET scan. So this is now what the clinic do right now. But I'm not a doctor, so I'm not familiar with what they really do in the clinical practice. I'm a chemist. So
Yeah,
I developed the
that even for experimental purposes, that's very, very interesting because yeah, for in terms of neurobiology, when we want to study the animal models, and it's possible to do some of these techniques in animals, it's nice to know that there are so many options now when we want to follow disease development, also from an experimental perspective. Thank you.
Yeah, thank you. Thank you.
Deji, do you want to ask your question again? Let's see if your connection is better now. Or perhaps not. Go to the Q&A. Danny, why don't you ask your question yourself there?
Yeah, sure. Thank you, Dr. Yan Cheng. Incredible talk, very beautiful work. And I believe my question is, it's mixed with Lauren's question that's in the Q&R. So my question is, do you believe that A beta protein is a step forward in Alzheimer's disease? We already know this. So do you believe that it be possible to develop other markers for other proteins using the same technique that you use for tau protein, for example, that you already said? Or Lawrence is asking for other markers like pH, local pH, redox potential, ionic strength, or something like that.
Yeah, that's very good question, and that's exactly what we are doing in my group. So we are developing. So there, how can I say? It actually it's not easy to develop tracers for NDDs because most of the biomarkers related to the pathogenesis is mainly happens in the brain. So all the tracers like the nanotechnology or something, we can't use that because we have to consider the brain uptake to how can they cross the BBB. So we mainly use the compact molecules as I introduced in the slides. So we have to use, and also the antibody is less used in the developmental tracer. So of course there's general procedure of how to develop the tracers, not only the radio tracers, but also the fluorescent tracers or photoacoustic tracers. They have the similar general procedures to develop. So of course use that, but the key point is not how to use the general procedure, but the key point is how to find a specific and selective tracer. So that's the key point, but that is very difficult. And besides that, we have to consider not only to consider the binding, they have specific and selective binding, but it also had to enter the brain effectively. And if it's intraneuronal, it also had to enter the neuron. So actually, there are so many barriers for the tracer to overcome. So that's a hard point. And also why the A beta like come first and approved in 20, like almost a decade before, because A beta is extracellular and it's very large size. So that's how can I say, it's easier. But for tau, it's intracellular. And also now what we are doing of the alpha synuclein is much harder because it's not only it's intracellular, and also they have smaller or lower concentrations in the brain. And with the coexistence of the A beta and tau, it's much harder because the tracer we developed, they have a basic molecule, like the selectivity is very important, or the issue for us chemists to develop a selective probe. So So yeah, we're still trying. We're still trying. Thank you for your question.
In the Q&A, we have a question from Lawrence Norris, and the question is, are there biophysical chemical markers that might lead to amyloid protein aggregation? For example, local pH, redox potential, ionic strength, etc.
Excuse me, I didn't really hear the question.
Are there other biophysical chemical markers that might lead to amyloid protein aggregation? For example, local pH, redox potential, ionic strength, etc.
For this question, the question is to consider the microenvironment, right? I'm not sure I get the question.
If you click at the top of the screen on Q&A, you should be able to see the question, and it's the first one from Lawrence Norris.
Yeah, I can't see the Q&A.
Oh, okay.
Okay. I saw that, Lawrence. Okay. There are other biophysical chemical markers that might lead to amyloid. Oh, yeah. Of course. The neurodegeneration is a very complex, multifactorial process. So, of course, the microenvironment in the brain, like it's mutual. It's mutual. So, of course, there are other markers that might lead to amyloid protein aggregation. So, what's very difficult for the development of the therapeutic intervention for Alzheimer's disease is because in the brain still we don't know much about the pathogenesis. So now we have a lot of hypotheses, and the primary one is the amyloid. So we developed a lot of anti-amyloid, but of course, besides the amyloid protein aggregation, there might be or maybe or should be some other kind of markers should lead to the amyloid protein aggregation, but we are not now for sure, or not enough as a biomarker directly related to the progression of the disease. I think diabetes plays. Oh yeah. For the second question, I think, yeah, of course, they have a lot of hypotheses. Of course, diabetes is one of them. Like as I mentioned in my talk, the COVID, people getting COVID now have the inflammation, and they then diagnosed with Alzheimer's disease. So maybe there's some connection. So they still need the, how can I say, research into that. Yeah, thank you for those questions.
Okay, and then we have a new question from Dr. Cheng. It says, from your talk, A-beta seems to be a good biomarker for disease and efficacy of treatment, but it is not a good target for drug intervention. What could be a potential good target for the drug intervention?
Yeah, that's also a very good question for the drug therapy for Alzheimer's disease. So for now, what I have known is that besides A-beta, they have found that tau is more related to the progression of Alzheimer's disease. So I think for the NDD, the treatment of NDD, the development of the tracers are very important, as they can provide the quantification tools to see if the intervention is effective, because it's very special because it's brain, so you cannot do the biopsies. Or so after we develop the tau tracers, so I believe that later there will be also anti-tau immunotherapies or likewise other immunotherapies for it, because we have the tools to see if there will be a reduction. Yeah.
Okay. There are no other questions in the chat. I see Dr. Balkis, you have your hand raised. You need to put your question into the Q&A. Give it a few minutes and see if anyone else puts a question into the Q&A. Okay, if there are no other questions, I just want to remind you that you can also, at the bottom of the slide on the screen, there's an email address. So if you do have some other questions, you can send it to that email address and we'll make sure that Dr. Cheng gets the question. Dr. Mahmoud, we can't answer your question that way. You have to put it into the chat or email it to pharmacologyeducation@elsevier.com. And since there's nothing else in the Q&A, then I'm going to end the session and say thank you very much. It was really a pleasure having you here, and we will be, within a week or two, we'll be uploading the recording of the presentation online so that it will be accessible to everyone. Thank you so much.
Thank you. Thank you all. Thank you.
Bye.
Bye.